Coming to the second week of intrauterine life or the second week of development, there are many changes that occur. So these are divided into changes that occur in the embryoblast, changes that occur in the trophoblast, and the changes in the uterine endometrium. So first we are going with the changes that occur in the embryoblast. You have the formation of the bilaminar germ disc. So easier to remember as it's the second week, so everything will be 2 okay. So you have the bilaminar, 2 layers in the germ disc. You have 2 cavities, 2 layers, chorion and amnion, like that you have to remember, okay. So coming to the formation of the bilaminar germ disc, bilaminar meaning 2 layers. Before we had the blastocyst looking like this. We had an outer trophoblastic layer and inner cell mass, okay. The outer layer or the outer cell mass that forms the trophoblast, and the inner cell mass that will it will form the embryoblast. Now what happens is the embryoblast, it will divide and it will form 2 layers, okay. It will form 2 layers. The above or the columnar layer, this one, that is called the epiblast, and the cuboidal or the layer below called the hypoblast, okay. The epiblast is of columnar cells, it is the primitive ectoderm. The hypoblast is of cuboidal cells, it forms the primitive endoderm. So now we'll come to this is the bilaminar germ disc. Now we'll come to the formation of the 2 cavities, okay. They can ask an MCQ, that is hypoblast is the first germ layer to be formed. Coming to the next cavities, what happens is, as these 2 layers from here, they will come over here and divide into cuboidal and columnar. What will happen is there is a gap formed over here. So in that cavity you will have a fluid called amniotic fluid or liquor alumni. It is amniotic fluid, okay. So in this cavity, since it is having amniotic fluid, it is called amniotic cavity. Now the amniotic cavity, its floor is lined by the epiblast cells, but its roof, so obviously you will have the trophoblast over here. So the trophoblast will divide and form one more layer over here that will form the roof of this cavity. So that layer is called amniogenic cells that originate from the trophoblast, not from the embryoblast but from the trophoblast. Then what will happen, again there will be a cavity below, okay. So here the hypoblast itself will divide like this. It will proliferate and it will form one cavity. This proliferated layer is called Users membrane. Users membrane, since it is from the hypoblast or the primitive endoderm, the Users membrane is endodermal in origin. So this cavity formed is called primary yolk sac. Primary yolk sac. So we missed one point over here before, is that the primary yolk sac, it forms on the 9th day. As on the 9th day, the hypoblast cells move this way and form the Users membrane. So it is formed on the 9th day. Coming to the formation of the extraembryonic mesoderm, the extraembryonic coelom, and the connecting stalk. So what we drew over here, this is the same structure. So you can see over here, this outer black lining is basically the trophoblast. The trophoblast then proliferates inward, okay. It proliferates inward to form the extraembryonic mesoderm. This entire thing is the extraembryonic mesoderm. It is also called as primary mesoderm because this is the first mesoderm formed. Now this part that you see, this is basically the embryo proper, and since this mesoderm is outside the embryo proper, it is called as extraembryonic mesoderm. After that you will also have intraembryonic mesoderm as we go down towards the next weeks. So here we have extraembryonic mesoderm. Coming to this, in this extraembryonic mesoderm, small cavities start forming, or small lacuna start forming. These lacuna then fuse to form one big cavity, as you can see in this picture. This one big cavity is the extraembryonic coelom. It is also called as the chorionic cavity, the extraembryonic coelom or the chorionic cavity. Now in this extraembryonic coelom, you can see due to this the extraembryonic mesoderm is over here and over here as well. So it is basically divided into two layers. The one more outside or the parietal layer is called as the somatopleuric extraembryonic mesoderm or the parietal extraembryonic mesoderm. It is also known as a chorionic plate. It is also known as a chorionic plate because it will form the chorion. Now you can see the more inner one or the more visceral one is called splanchno-pleuric extraembryonic mesoderm or the visceral extraembryonic mesoderm. As you can also see over here, due to the formation of this cavity, actually the secondary yolk sac, the primary yolk sac will shorten or it will become smaller like this. It will be pushed by this cavity. So it will now form the secondary yolk sac. The primary yolk sac will become smaller and will form the secondary yolk sac. Now this extra embryonic coelom, what will happen is it will not extend fully. It will not extend where the extra embryonic mesoderm connects the amniotic cavity to the trophoblast. It will not extend there. So this part where there is no cavity, where there is actually extra embryonic mesoderm, this part will in future form the future connecting stalk. And this future connecting stalk will in turn down the lane form the umbilical cord. Okay. Now with this division of the extraembryonic mesoderm, you have two layers formed again. One is the amnion, the other is the chorion. So we'll see what those are. So as you can see over here, the chorion is basically the trophoblast. The trophoblast will actually form two layers. I will see it in the next part. That is the syncytial trophoblast and the cytotrophoblast. This part, these three layers, that is the syncytial trophoblast, cytotrophoblast and the somatopleuric extraembryonic mesoderm will together form the chorion. And then for the amnion, you have this two layers, the amniogenic cells and the splanchno-pleuric extraembryonic mesoderm. Coming to the formation of the procaudal plate. Okay, so before what we saw, this was the embryo proper that we were seeing. It was actually circular in structure, so it is called as the embryonic disc. Okay, this embryo proper consisted of the epiblast and the hypoblast cells. They were arranged like this. Above and below, epiblast and hypoblast, they were circular in nature, forming the embryonic disc. So now we already knew that the cells of the hypoblast are cuboidal. Okay, on day 14, at one part these cells will become columnar. This part where the cells become the columnar will form the procaudal plate, as you can see over here. So in this area, the cells of the hypoblast have become columnar. This is called the procaudal plate. So this procaudal plate is the first indication of the cephalic and the caudal end, or it is the first indication of a direction in the embryo. Okay, before this, since it was circular, there was no indication of head end or tail end or anything. But now with the procaudal plate, you can see this part will be the head end or the cephalic end. This part will be the tail end or the caudal end. This formation of the procaudal plate will give you the cephalocaudal axis or the vertical axis or the head-tail axis. Okay. At the procaudal plate, the cells of the epiblast and the hypoblast are adherent tightly. They will be like this only. They will be adherent tightly to each other even during the third week. In the third week, you will actually have the formation of the intraembryonic mesoderm that will separate these two layers. That will separate the epiblast and the hypoblast. But at this procaudal plate, these cells will be tightly arranged. They will be adherent to each other. That is, the intraembryonic mesoderm cannot come between these two at the procaudal plate. So this part is also important further down the lane. Coming to the changes that occur in the trophoblast. So we have already seen that the embryoblast divides into the epiblast and the hypoblast on the 8th day. Similarly, the trophoblast also, on the 8th day, it will divide into two layers. The syncytial trophoblast, that is present towards outside. Now, why is it called syncytial? Now, in this layer, the cells, they lose their walls and they fuse with each other. This fusing with each other forms multinuclear structure. A multinuclear structure or a syncytium. Thus, it is called syncytial trophoblast. It is also known as plasmodiotrophoblast, again because of its multinuclear structure. Coming to the cytotrophoblast, it is present towards the inside, and cyto because the cells over here are clear and distinctly separated. As they retain their walls, they are mononuclear. This is also called as Langerhans layer. Langerhans layer. Coming to the decidua. Now this actually is a part of the first week, but is important to understand. It is important to understand this to understand the second week. So coming to the decidua, it forms on the 5th day. The decidua is a functional and specialized endometrial lining or the stratum compactum of uterus during pregnancy after implantation. This supports the embryonic development. So basically it is the endometrial lining, the stratum compactum, after implantation occurs. Okay. So if you see this, the decidua has three divisions. Okay, the first one is the decidua basalis, then you have the decidua capsularis, and then you have the decidua parietalis. So the decidua basalis is adjacent to the implanted blastocyst. So this part of the decidua that is adjacent to the implanted blastocyst is called the decidua basalis. It forms the maternal part of the placenta. Here the maternal blood vessels, they proliferate and then they dilate to form sinusoids, because in the future this will form the placenta, right? So here you need more blood supply. So then the next part, you have the decidua capsularis, the endometrium surrounding the conceptus. So this is the conceptus. Endometrium surrounding the conceptus, this part is called the decidua capsularis. Decidua capsularis. Now what will happen as this conceptus or the embryo, as it grows in size, it will push this layer. It will push this layer and it will stress it. So about third month where it grows too big, this decidua capsularis will come and fuse with this layer. So this cavity that you see in between, that is the uterine lumen or the uterine cavity. Okay. The last layer is the decidua parietalis. That is the rest of the decidua lining the uterine cavity. So this much, or the rest of the decidua that is not lining the conceptus, is the decidua parietalis. In the third month, as this embryo grows too much, the decidua capsularis will come over here and it will fuse with the decidua parietalis and it will obliterate the uterine lumen. So there will be no uterine lumen anymore.